Hello everyone, Doc Ian here. This is the part 4 of our lecture on Introduction to Immunology. Immune responses are not generated at the same time. They are produced in sequences. How the immune system will determine the sequence of immune responses is highly comparable with how we play the chess game. We usually start the game by moving the pawns because we can afford to lose them rather than losing the major chess pieces or characters. That is also how the immune system will send its components to fight off the infection. It will start first with certain types of cells. If these cells will undergo apoptosis, or if let's say they will not work, these cells will now be replaced by another group of cells. That's why immune responses are divided into first line, the first one that will come in contact with a pathogen or offending microorganism. And then we have the second line of defense. Then we have the last one, the major, the third line of defenses. Immune responses in our body are divided into first line, second line, and third line of defenses. Please always remember that the first line of defense or that group of immune responses will always prevent the entry of microorganisms in our body, particularly to our blood circulation. If the first line will fail, the immune responses that will start immediately will belong to the second line of defense. For you to understand that one, I want you to imagine the skin. The skin is part of the first line. It will always prevent the entry of microorganism. Should there be an injury resulting to a wound, the skin at this point will no longer be able to provide first line of defense. But what will you notice? The area where there is the injury, there would be what? Inflammation. Inflammation now will try to localize the infection, and inflammation is part of the second line of defense. So what is now the third line of defense? The third line of defense would involve the actions of your T cells and B cells. So therefore, third line of defense belongs to the adaptive or specific immunity, while the first and the second lines of defenses are classified under non-specific or innate immunity. Thanks to God, because our first two lines of defenses are non-specific, they will not select what to protect us from, and they are already available upon birth because they are part of the innate immunity. Again, the immune responses that belong to the first line of defense would always prevent the entry of microorganisms in our body, particularly in our blood circulation. Why do I have to always emphasize that the organism should not reach our blood circulation? I will explain that later on. The first line of defense is further divided into anatomical, chemical, and mechanical. In terms of anatomical, we have the following examples. The skin, which is able to provide us a good barrier against the entry of microorganisms. The sweat glands, which can produce sweat, and sweat is high in salt content. And I want you to review your knowledge on physiology. If cells are placed in hypertonic solution, they will what? Shrink. And shrinking of cells can cause their death. So if you have bacteria on your skin, but if they will be exposed to sweat, then most likely they will undergo cell shrinkage. Next, sebaceous glands, which are responsible for the production of sebum. How do you think sebum can contribute to immune responses? Sebum is high in fatty acids. Fatty acids. And anything acidic can actually kill microorganisms. Lacrimal glands are also classified under anatomical first line of defense because lacrimal glands can produce tears, and tears are rich in lysozyme. And you have learned from the part 1 of Introduction to Immunology that lysozyme can damage bacterial cell wall. Another classification of first line of defense is chemical. So the examples are the hydrochloric acid in the stomach, which can kill bacteria ingested along with our food. The reason why I have to emphasize that first line of defense will prevent the entry of microorganism, especially up to the blood circulation, is that most of you would think that hydrochloric in the stomach belongs to second line. Why? Because the hydrochloric acid is already found inside the body. Because, di ba, the way we defined first line is it's supposed to not allow the entry of microorganism in the body. And the bacteria and the food has already entered the body. So to emphasize that first line would involve hydrochloric acid in the stomach, the lactic acid in the vagina, the mucus in the respiratory tract. I have to emphasize that what I mean by prevention of entry to the body is that the organism must not be able to reach the blood circulation. Another example of chemical first line of defense is the lactic acid produced by the Lactobacillus acidophilus in the vagina. That's why in some disorders affecting the vagina, the problem starts with what? The loss of the acidity in the vagina, secondary to the death of the Lactobacillus acidophilus. Lysozymes in tears, which can break down bacterial cell wall. The mucus in the respiratory tract, which can trap foreign objects such as bacteria and other microorganisms present in the air that is passing through the airway. And then the sweat, which has high salt content, and as well as the sebum, which has fatty acids. The third group of first line of defenses are classified as the mechanical, and the examples are the following: sneezing and coughing, because every time we sneeze, every time we cough, we are removing something that is irritating our airways, and that's one of our ways on how to prevent the entry of microorganisms in our body. And this one can also include vomiting, especially if we have ingested microorganisms or toxins along with the food. Next, urination and defecation. Why? Because every time we urinate, we are actually reducing the number of microorganisms in our urinary tract. And same is true with defecation. So if your mother is already knocking at the door of the CR because it's taking you so long to, defecate, you have to answer her, "Ma, please wait, I'm still in the middle of a fight." Because you are reducing the number of microorganisms in your gastrointestinal tract. We have already finished discussing the first line of defenses. It's now time to go to the second line of defenses. Take note, if the microorganism will escape or evade the first line of defense, what will come into play is the second line of defenses. First line and second lines of defenses fall under non-specific immunity. First line will always prevent entry of microorganisms in the body. If it will fail, you will now have the second line of defenses. These are the immune responses that are classified under second line of defenses. Let us start with inflammation. If the skin will be injured, the skin is no longer providing us the first line of defense. So what will set into play is the inflammation. That's why you will notice the injured site of the skin will become swollen. We will discuss the second lines of defenses one by one, but I will no longer discuss the activity of natural killer cells because we have already emphasized the function and the actions of natural killer cells in our previous lectures. Every time that our skin will have injury or wound, we will notice that the affected site will become inflamed. And the main purpose of the inflammation is to localize the infection so that the introduced microorganisms will not be able to spread to other areas in the body. Inflammation is defined as a local response of human tissue to injury. And again, one of its purpose is to localize the infection. And while localizing the infection, it will also try to eliminate the cause of injury or the organisms introduced along with the injury. Next, since the area is contained, it will also allow some of the cells to do some tissue repair. So from now on, every time you will see inflamed areas in the bodies of your patients or even on your own body, you always have to be reminded that the immune system is trying to contain the infection and it's trying to eliminate the cause of injury while allowing the tissue to also undergo repair. Inflammation falls under second line of defense because this will never happen if the skin has not failed in providing the first line of defense. I know this one is already familiar with you because you have already learned this in histopath. Let's have a review. These are the cardinal signs of acute inflammation. Should there be any injury, you will notice that the injured site will have redness and heat. Redness is termed rubor. Heat is termed calor. The color of the gem ruby is red. So it's easy to memorize that rubor means redness. Every time you go to the gym and do some workouts, you're actually burning some calories. When you burn, there's heat, calor. You will also notice that the injured site will become swollen, and the term that would refer to it is tumor. And the pain brought about by the injury is termed dolor. In some injured or inflamed tissues, they may not be able to regain their original function. That's why there is loss of function. At this point, we will try to discuss the mechanisms behind the cardinal signs of acute inflammation. So as you can see in the picture, you have here a sharp object piercing through the skin and introducing microorganisms. I want you to take a look at the blood vessel immediately below the injured site. Can you appreciate that after the injury, the blood vessel has dilated? And as you can see on the second picture, you have there the movement of fluid from the lumen of the blood vessel into the injured tissue. The mechanism behind the redness and heat, or rubor and calor, is the release of histamine, bradykinin, and prostaglandin from the damaged cells. I hope you can recall that when the sharp object pierced through the skin, it has caused damage among the cells present in the epithelium and connective tissue. These damaged cells will release the chemicals histamine, bradykinin, and prostaglandin. One of the physiologic effects of these chemicals is vasodilation. As shown on the previous slide, you've noticed that the blood vessel has increased its diameter, and that's vasodilation. With vasodilation, there is increased blood flow. When there is increased blood flow, there would be more RBCs reaching the site of infection, and RBCs have hemoglobin, and hemoglobin imparts red color, and that's the reason why there is redness. There is heat because when there is increased blood flow, there would also be increased flow of heat towards the site of infection. As the sharp object pierced through the skin, the damaged cells release histamine, prostaglandin, and bradykinin, the ones represented by the blue circles in the photo. And these chemicals now will cause the capillaries or blood vessels to dilate. That's why in number 2, you can see that the blood vessel dilated, allowing now increased blood flow to the area, contributing to the rubor and calor. Again, increased blood flow will lead to increased amount of blood reaching the site of injury, and then that will now result to heat and redness. Another physiologic effect of histamine, bradykinin, and prostaglandin is increased vascular permeability. These chemicals can cause the endothelial cells, the simple squamous epithelium of the blood vessel, to contract. If they will contract, they will have spaces between them. And when they do, the spaces now will allow fluids to leak out of the blood vessel. Take a look at the simple squamous epithelium of the blood vessel in the photo. Can you appreciate that in the middle part of the photo, the endothelial cells became shortened? It's because they were contracting, and that's their response to histamine, prostaglandin, and bradykinin. If these cells will contract, they will have gaps between them, and the gaps now will allow the escape of fluids into the site of injury, and that is now increased vascular permeability. And if there's escape of fluid, the fluid will accumulate, and that will now cause the injured site to swell. And that is now the reason behind the cardinal sign tumor. What do you think is the reason for allowing these fluids to leak out? When fluids leak out, they carry along with them antibodies. And antibodies can help in what? Localizing the infection and eliminating the cause of injury. And when endothelial cells contract, they would have gaps between them, and the gaps will allow the white blood cells to go out and go to the site of injury and eliminate the offending microorganism. So I want you to watch this animated clip. You have to hear the release of bradykinin and histamine. And what do we notice? As they are released, they will later on cause the leakage of fluid from the blood vessels, resulting to the swelling of the injured site. The fourth cardinal sign is pain or dolor. And this is primarily because of the action of these mediators, chemical mediators, the histamine, bradykinin, and prostaglandin on the nerves supplying the injured site. So I want you to take a look at this picture. On the left side, you have there the injured site, and you have there the release of bradykinin, histamine, and prostaglandin. And these chemicals will act on the nerve endings. And these nerve endings now will be activated, and they will send nerve impulses to the brain, and the brain will interpret it as pain. Bradykinin, histamine, and prostaglandin will activate peripheral nerves, and these nerves will send the stimuli to the brain, which will interpret the stimuli as pain. The area where there is injury will become inflamed, and for the organisms present on the inflamed area to be eliminated, we need white blood cells to go out of the blood vessel and go to the site of injury. And the migration of the white blood cells out from the blood vessel towards the site of injury is termed chemotaxis. And chemotaxis will now make it possible for the white blood cell to engulf the foreign material or the offending microorganism in the process called phagocytosis. And that is our second example of immune responses that fall under second line of defenses. Again, chemotaxis is the movement of the white blood cell from the blood vessel towards the site of infection. And chemotaxis has 4 important steps. Please take note that the white blood cells usually circulate in the central portion of the blood. If they want to go out because they are attracted to the chemotactic stimuli, they have to undergo 4 important processes for the chemotaxis to be completed. It starts with, let's wait for it, margination, followed by rolling, and then adhesion, then the transmigration or diapedesis. Chemotaxis has 4 important stages. I hope you have already memorized them. Margination, rolling, adhesion, and the diapedesis or transmigration. In the early stage of the inflammation, there are still few microorganisms present. With their number, they will only be able to produce low level of chemotactic stimuli. And this low level of chemotactic stimuli will start attracting the white blood cell that is most sensitive to chemotactic stimuli, and they are the neutrophils, right? Okay. So, what will happen is, from the central portion of the blood, the white blood cell will migrate towards the endothelium or the wall of the blood vessel. So, it will go from the central to the margin. That's why the step or the process is called margination. I want you to imagine as the white blood cell is traveling or migrating from the central portion of the blood vessel towards the margin or the wall of the blood vessel, this white blood cell will also start expressing on its surface L-selectin. Again, imagine in a normal person or a healthy person, the white blood cells are most of the time found in the center. when there is low level of chemotactic stimuli, they will start to go to the margin, and you call that as margination. As they are undergoing margination, they will start expressing on their surface or cell membrane L-selectin. And the endothelial cells, the simple squamous epithelium of the blood vessel, will also express E and P selectin. What's the purpose of the white blood cells to have L-selectin and the endothelial cells of the wall of the blood vessel to express E or P selectin? Remember, the blood is continuously flowing. If the white blood cell will go to the margin and will not have something to anchor onto, the white blood cell will be pushed by the cells behind it back to the central portion of the blood. And none of these white blood cells will be able to go out of the blood vessel and go to the injured site. So the L-selectin will enable the white blood cell to anchor itself or attach itself to the wall so that it will no longer be carried back to the central portion of the blood. And the E-selectin will allow the white blood cell to do that because the E-selectin will bind to the L-selectin of the white blood cell. But the thing is, the binding between L and E is weak. So eventually, the white blood cell will detach its L-selectin from the E or P-selectin of the endothelium. Another thing, L-selectin, white blood cell, because white blood cells are known as leukocytes, L-selectin, leukocyte. E or P-selectin is present in endothelium, E-selectin, endothelium. The L-selectin will enable the white blood cell to attach itself to the simple squamous epithelium or the wall of the blood vessel. But the binding of L-selectin to the E-selectin of the endothelial cell is weak. So eventually, the white blood cell will detach or the phagocyte will detach from the wall. But do not worry about it. The L-selectin is distributed all throughout the white blood cell, and the E-selectins are also distributed all throughout the length of the blood vessel wall. So what will happen? The white blood cell will just undergo the next step of chemotaxis, which is what? Rolling. So rolling is mediated by L-selectin of the white blood cell and the E and P selectins of the endothelial cells. As time goes by, the number of microorganisms will increase because these organisms will replicate. With the number, the expected that the chemotactic stimuli level will also increase. If the chemotactic stimuli will increase, there would be a sudden change in the expressed proteins or receptors on the surface of the white blood cell and the endothelial cell. Please take note that the L-selectins will be replaced by integrin, while the E and P selectins of the endothelium will be replaced by ICAM. Higher levels of chemotactic stimuli will cause the white blood cell to shift from expressing L-selectin to integrin, and the endothelial cells will shift from expressing E or P selectin to intercellular adhesion molecule or ICAM. The binding of integrin to ICAM is very strong. That's why you should not expect the white blood cell to roll this time. The white blood cell will tightly adhere to the endothelial cell. That's why the third step is called adhesion. And the squeezing through of the white blood cell between two endothelial cells is now called as diapedesis or transmigration. And the protein involved in diapedesis or transmigration is PCAM. So take a look at this white blood cell. As you can see, it will squeeze itself through the gaps between two endothelial cells. And you call this process as diapedesis or transmigration. Take a look at the pictures of these three patients. What do you notice? They have areas in their body that are highly infected or inflamed. And with this severity of infection, expected that these areas should present with pus or purulent discharges. But looking at these pictures, you don't see any purulent discharge or pus. These patients are suffering from what we call leukocyte adhesion defect, wherein some of the proteins involved in the 4 steps of chemotaxis are not encoded by their genes. If those proteins will go missing, the entire process of chemotaxis will not be completed. Therefore, the neutrophils will not be able to go out of the blood vessel and go to the site of injury. That's why you don't see any pus in there, because in the first place, the neutrophils failed to reach the site of infection and become activated and die to form the pus. There are 2 types of leukocyte adhesion defect, and I want you to listen very carefully. Type 1 and type 2. Type 1 is the absence of integrin. Leukocyte adhesion defect type 1 is the absence of integrin. So what process of chemotaxis is affected? Integrin what? Adhesion. Very good. Leukocyte adhesion defect type 2 is characterized by the absence of L-selectin. So what step in chemotaxis do you think will be greatly affected? The answer is rolling. Absence of any of the 2, integrin or L-selectin, will most likely present with this clinical manifestation. The migration of the white blood cell out of the blood vessel towards the site of infection is termed chemotaxis. Once the white blood cell is already out of the blood vessel and is about to engulf the offending microorganism, you will call the process as phagocytosis. Phagocytosis is defined as the process of engulfment of solid particles such as bacteria and yeast cells by our white blood cells. Phagocytosis involves 3 steps. You have recognition and attachment, you have engulfment, and you have digestion. And we will discuss them one by one. After completing the entire process of chemotaxis, the white blood cell is expected to go out of the blood vessel. After that, the white blood cell might bump into a healthy human cell and the microorganism. So the next question now is, the phagocyte doesn't have a brain. It doesn't have eyes. How will the phagocyte now know which among the cells is an enemy or a friend? So how will the phagocyte decide what to eat and what not to eat? To answer that, I want you to learn the concept on pathogen-associated molecular patterns, or simply known as PAMP. What is a PAMP? A PAMP is any molecule that is shared by most microbes or most microorganisms, and these PAMPs play important role in their survival. But take note, these molecules are not present in our body and in our own cells. So if the neutrophil or any white blood cell will detect the presence of a PAMP, that will give the white blood cell the clue. The cell in front of it is not from the body and it's supposed to be eliminated. So this PAMP now will tag the microorganism as foreign, therefore it should be attacked by the white blood cell. Cells with PAMPs are considered as foreign, thus they should be engulfed by phagocytes. So take a look at the picture of the phagocyte. Can you appreciate that it has green-colored receptors on its surface? And the main purpose of these receptors is to detect the presence of PAMPs, such as what? Lipopeptide, flagellin, peptidoglycan. The 3 mentioned chemicals are not present in our cells, so therefore they are considered to be PAMPs. The next question now is, what do you call the green-colored structures that are used by the phagocyte to determine whether the cell in front of it has a PAMP or not? These receptors are called pattern recognition receptors, or PRR. So PRRs are found in our phagocytes, and PRRs are supposed to identify PAMPs. So take a look at this photo. The yellow colored structure there is the phagocyte, and then displayed on its surface are the pink colored PRRs, and then you have there the red colored bacterium, and then expressed on the surface of the red colored bacterium are the golden colored PAMP or PAMPs. So the phagocyte will use the pink colored PRRs to detect the presence of the golden colored PAMPs on the bacterium. If the phagocyte will now detect the presence of PAMP in a cell, then that cell with the PAMP should be engulfed by the phagocyte. But some bacteria has this covering we call capsule. And capsule will help bacteria escape phagocytosis. So I want you to take a look at the picture of the phagocyte. The phagocyte is about to engulf a bacterium, but that bacterium contains capsule. And the capsule somehow will make the bacterium slimy, and they will just slip off from the site of engulfment. Thereby, bacteria with capsule are difficult to kill via phagocytosis because they have something that will make them evade phagocytosis. So, how do you think the phagocytes will still engulf capsulated bacteria? If you have something slimy and you want to pick it up and you cannot do so because it's slipping off of your hand, you will try to cover it first with a tissue paper so that the tissue paper now will hold on to the sticky material, and you will be able to pick up now the sticky material by using the tissue paper. We will also try to apply that one in our phagocytes. The phagocytes will first coat these capsulated bacteria with substances which will make them less slimy so that it would be easy for the phagocytes to engulf them. The process of coating antigens with chemicals that will enhance the process of phagocytosis is called opsonization. Opsonization. And there are 3 possible substances that can facilitate opsonization. So I want you to memorize them by heart. You have the antibody IgG, you have C3b, and you have CRP. Please don't proceed to the next slide or the remaining parts of this recorded video without you memorizing the 3 types of opsonins. Again, IgG, C3b, and CRP. So I want you to take a look at the pink-colored bacterium in the photo. That bacterium contains capsule. But since the bacterium is covered by IgG, the green-colored Y-shaped structure in there, or figure, the IgG now has opsonized already the bacterium. And it would now be easy for the yellow-colored phagocyte to engulf the capsulated bacterium. So take a look at the phagocyte. It's about to engulf a capsulated bacterium, but this phagocyte will have a hard time doing that. But if the bacterium is covered by those letter C's in there, those letter C's are representing the C3b, one of our opsonins, the C3b will bind to the red-colored C3b receptors present on the membrane of the phagocyte. And what will happen is after the C3b will bind to the C3b receptor, that will now result to the engulfment of the opsonized bacterium. Letter C is showing us a bacterium coated with IgG, one of our opsonins. And since the FC portion or the tail portion of antibodies have corresponding receptors on the membrane of the phagocyte, then the opsonized bacterium will later end up getting phagocytosed. After recognizing that the cell in front of it is food and it has PAMP, it's now time for the phagocyte to engulf the pathogen or offending microorganism. So the next step for phagocytosis is engulfment. Engulfment is done by forming a membrane fold around the particle or bacteria. And this structure will later closed and form a vesicle. So I want you to take a look at the photo. Can you appreciate that there's a membrane fold trying to surround the bacterium, and that fold will later fuse together, and the fusion will cause the formation of the vesicle, the one shown in the letter C part of the picture. And that vesicle now will be called as a phagosome. The phagosome functions to contain the engulfed material. I want you to realize that if that bacterium is placed directly in the cytoplasm, the enzymes in the cytoplasm will kill the bacterium in an instant. So the next question is, why do we have to store the bacterium inside the phagosome if it will die instantly in the cytoplasm? There are bacteria that can produce toxins, and the toxins can kill the phagocytes. So the phagocytes have to protect themselves from the toxins of the bacteria. So for the toxins not to kill them, might as well keep the bacteria inside phagosome, so that if the bacteria will produce toxins, the toxins will just be released inside the phagosome and will not cause the death of the phagocyte. You see how intelligent our phagocytes are. They know how to protect themselves. They know how to avoid getting hurt. Ikaw na may utak, alam mo nang nasasaktan ka, go nang go ka pa rin. So shown in here is the process of engulfment. You have there the formation of the membrane fold that will later surround the bacterium, and the fold will close to form the phagosome. So let's apply what you have learned so far. You have here in number 1 the microbe or the particle, and in number 2 you can see you have there the process of engulfment. You have there a membrane fold trying to surround the bacterium, and if the ends of the fold will fuse together, you will now have the formation of the phagosome as shown in number 3. The bacterium inside the phagosome has to be killed. It has to be digested. That's why the third step in phagocytosis is the digestion. And digestion is made possible by the organelle lysosome. So, I want you to take a look at number 3. You have there the phagosome with the enclosed bacterium. And for the bacterium to be digested, the phagosome will fuse with the organelle that has the powerful digestive enzymes within its compartment, and that is the lysosome. Can you appreciate in number 4 that the lysosome will fuse with the phagosome, and they will form a phagolysosome. And the main purpose of the phagolysosome is to initiate the digestion of the bacterium. Once digested, the residual body or the waste product will just be released outside the cell in the process called exocytosis. There's one disease that I want you all to take note of, and that is Chediak-Higashi Syndrome. Chediak-Higashi Syndrome is characterized by the absence of the lysosomal trafficking protein or LYST protein. What is the importance of this protein? This protein is responsible for making the phagosome fused with the lysosome. If this protein is absent, then the fusion of lysosome and phagosome will not happen. That's why children born with Chediak-Higashi syndrome will have recurrent bacterial infections because their phagocytes cannot kill the bacteria. Do you know that patients with Chediak-Higashi will present with albinism? Why? Take a look at the photo on the left side of the screen. Can you appreciate that you have there the epidermis? And take a look at the lowermost layer of the epidermis. The epidermis is colored pink in the picture, by the way. The lowermost layer of the epidermis is called stratum basale. And it is in the stratum basale where you can find the cell responsible for the production of the pigment melanin, and that's the melanocyte. The melanocyte is color brown in the picture. This melanocyte has to share the melanin pigment to the other cells of the epidermis. And what will help these melanocytes distribute the melanin to the keratinocytes and make the skin appear brown is lysosomal trafficking protein, the same protein involved in the fusion of lysosome and phagosome. So for a person with Chediak-Higashi, fusion of phagosome and lysosome will not happen, and the distribution of melanin from the melanocyte to the other cells of the epidermis will not also occur. That's why people with Chediak-Higashi syndrome will manifest with albinism and recurrent bacterial infections. Bacteria can be classified as aerobic or anaerobic. They are classified as aerobic if they need oxygen for survival. They are classified as anaerobic if they consider oxygen as toxic to them. So therefore, these organisms prefer to grow in areas where there is no oxygen present. Phagocytes are specialized in killing aerobic bacteria. They will actually offer this bacteria oxygen. So I know you are reacting, why offer oxygen to bacteria that need oxygen for them to survive? These phagocytes will offer oxygen, but they will make it a point that the oxygen is toxic or poisonous to the bacteria. The bacteria will accept the oxygen without question, and they will use the oxygen, and without them knowing, the oxygen can kill them. Now, how do you think the phagocytes can come up with the toxic oxygen radicals? They will first make oxygen react with NADPH. You have to memorize the chemical reactions. The chemical reaction, the first chemical reaction is mediated, facilitated by the enzyme NADPH oxidase. NADPH oxidase. Although the one written in the PowerPoint is phagocyte oxidase. But I think NADPH is easier to memorize because your substrates are NADPH and oxygen. The product of the chemical reaction is superoxide. Superoxide is already considered as a toxic oxygen radical, but it's not as potent as the other toxic oxygen radicals. So if this is the one that will be offered to the aerobic bacteria, some of them will die, but majority of them will not. So there's a need for the superoxide to be converted to a more potent toxic oxygen radical, and that's hydrogen peroxide. And that is made possible by the enzyme superoxide dismutase. Again, NADPH plus oxygen with the action of the enzyme NADPH oxidase will produce superoxide. Superoxide will be acted upon by superoxide dismutase, then you will have hydrogen peroxide. Do you know that these chemical reactions are the principle behind the discovery of the agua oxinada? Agua oxinada primarily contains hydrogen peroxide. And diba, you're supposed to pour it on your wounds, and then you will see bubble formation, and that would only indicate that the toxic oxygen radical in the form of hydrogen peroxide is doing its killing activity. The problem with hydrogen peroxide, yes, it is potent, but the thing is there are bacteria that can express the enzyme catalase. If the bacteria have catalase, the catalase will help the bacteria break down hydrogen peroxide into water and oxygen, and the toxic oxygen radical will not be able to kill the aerobic bacteria. That's why hydrogen peroxide should be further acted upon by myeloperoxidase, converting it into hypochlorite, one of the main components of what we know as the commercial product named Zonrox. Let me introduce you to another primary immunodeficiency disorder. We have the chronic granulomatous disease. Chronic granulomatous disease is characterized by the absence of NADPH oxidase. And you know very well how important this enzyme is. It converts oxygen to superoxide, and superoxide can be later converted to hydrogen peroxide. Without this enzyme, the phagocyte will not be able to, or the ability of the phagocyte to kill aerobic bacteria will most likely become impaired. As for the second lines of defenses, we have already covered inflammation, then phagocytosis. We have discussed under phagocytosis chemotaxis. We will now go to our second line of defense. Fever. Is fever an immune response? Most of the bacteria that are causing us to get sick are classified as mesophiles. And take a look at the growth curve of mesophiles. Can you appreciate that their growth will peak at 37 degrees Celsius? And that is our body temperature. So every time that bacteria will enter our body, the temperature inside will be optimum for them to replicate. But our body is wise. Since our body knows that they need 37 degrees Celsius for them to replicate, the body will make the environment inside unfavorable to them. The body will increase the body temperature to 38, 39, and 40. And I want you to appreciate the drop in the growth curve of mesophiles if the temperature will be increased. And that is now the purpose of fever. What is the mechanism behind the occurrence of fever? I want you to take a look at the yellow-colored macrophage in the photo. And as you can see, it has presented an antigen using class 2 to T helper cell. Once the T cell will be activated by the antigen presentation, the macrophage or the APC and the T helper cell will detach from each other. The activated T helper cell will then look for the B cell that is specific to the antigen presented by the antigen presenting cell or the macrophage. I want you all to understand that from the time that you will be exposed to the infectious agent up to the time that the infectious agent will be presented by the antigen presenting cell to the T helper, up to the time that the T helper will look for the B cell, until the time that the B cell will have to transform first to become the plasma cell, and until the time that the plasma cell will be able to produce antibodies, you have that certain period, minimum of 7 days, that you don't have antibodies because they do not appear immediately after exposure to the infectious agent. That's why there is that minimum of 7 days that you are left unprotected because you are still waiting for the antibodies. So your immune system must come up with immune responses that will somehow protect you while waiting for the antibodies to appear. That's why after the detachment of the antigen presenting cell or the macrophage from the activated T helper cell, the macrophage will have to do something. You see, the role of the macrophage does not end up with antigen presentation. After detaching from the activated T helper cell, this antigen presenting cell will produce 3 important cytokines. We have interleukin 1, you have 2 more necrosis factor, then you have interleukin 6. What do you think will be the physiologic effects of these cytokines? Let's discuss them one by one. You have here a picture of a macrophage, and let's assume that this macrophage has already presented the antigen to the T helper cell. This macrophage now, after it will detach from the T helper cell, will produce the interleukin 1, the one in the red rectangle in the photo. This interleukin 1 will circulate in the blood and will go to the hypothalamus. Hypothalamus sets our body temperature. If the hypothalamus will detect the presence of interleukin 1, it will reset our basal body temperature. So it will increase our body temperature, and that will now be referred to as fever. And the main purpose of the fever is to not allow the organisms to have favorable environment for them to replicate. So, the next time you will develop fever and your mom will ask you, "What's wrong with you, anak?" You look into her eye and say, "Ma, I am fighting." Because fever is an immune response. So, I want you to change your idea about fever. From now on, do not treat fever as a disease. It's actually an immune response against infectious agents. The next immune response that falls under the second line of defenses is the production of acute phase reactants. The production of these chemicals are somehow related to the actions of the cytokines produced by the antigen-presenting cell or the macrophage after detaching from the activated T helper cell. Take a look at the macrophage in the picture. Assume that this macrophage has just recently detached from the activated T helper cell after the antigen presentation. This macrophage, as shown in the picture, has synthesized interleukin-1, interleukin-6, and 2 more necrosis factor. These 3 cytokines will act on the liver. And the response of the liver is to produce sets of proteins. Remember, it would take days before the antibodies will appear after you are exposed to the antigen. So, what will temporarily act as quote-unquote antibodies and provide you protection while you're waiting for the real antibodies are the proteins that will come from the liver. And these proteins now are called acute phase reactants. That's why if the levels of these acute phase reactants are high in your blood, that would only indicate that you're having acute inflammation. Acute phase reactants are proteins produced by the liver during the acute stage of the infection. And this is in response of the liver to the production of interleukin-1, interleukin-6, and 2 more necrosis factor by the phagocytes. And these are the examples of acute phase reactants. Lactoferrin. Lactoferrin is actually a iron transporting protein. So most of you would wonder, what is now the importance of lactoferrin in generating immune responses against bacteria? Lactoferrin transports iron. If lactoferrin is present in high amount, they can deprive the bacteria of iron by transporting them away from the bacteria. And the bacteria now will not have iron molecules to use, and that could cause them to die. Alpha-1 antitrypsin is produced by the liver in high amount in acute stage of infection. Why? Because in acute stage of infection, neutrophils are actively clearing bacteria. And when they try to clear bacteria, when they try to engulf bacteria, their elastase enzyme might escape from their granules and destroy tissue. So might as well have something that will neutralize the elastase, and that is alpha-1 antitrypsin. Ceruloplasmin is a copper transport protein. So therefore, it will deprive the bacteria of copper, and that can result to their death. Fibrinogen is also an acute phase reactant. And if this fibrinogen is activated to form fibrin, it will form clots, and the clots now will trap bacteria. So you see, these proteins may not have the same activities as the antibodies, but they can somehow provide protection up to some point. You also have C-reactive protein as part of the acute phase reactant. And you know very well that CRP is one of the opsonins. CRP stands for what? C-reactive protein. And another member of the acute phase reactants is the complement proteins. But we will have a separate discussion on this type of proteins in week number 5. Another immune response that falls under the second line of defense is interferon, particularly the alpha and the beta interferon. Take a look at these 2 cells. The cell on the left side of the screen is infected by a virus. And since this cell doesn't want the cell beside it to be infected by the same virus, the first cell or the virally infected cell will produce a chemical. And that chemical is called interferon. And that's the one shown on the lower part of the cells. Can you appreciate that the virally infected cell released alpha and beta interferon, and the alpha and beta interferon diffused towards the neighboring cell. And then what will happen is, once the virus focuses on the upper parts of the cells, can you appreciate that the virus is about to transfer from cell number 1 to cell number 2? Since cell number 2 is already activated by interferon, the virus now cannot anymore establish infection on cell number 2. The infection was interfered That's why the chemical is called interferon. So interferon is produced by virally infected cells and can somehow provide protection against viruses. Since interferons, alpha and beta, will not choose what virus to protect us from, then therefore, these interferons alpha and beta fall under non-specific immunity. And since they are part of the second line, you have learned a while ago that second line of defense falls under non-specific or innate immunity. These are the different types of interferons. We have interferon alpha and interferon beta. Interferon alpha is produced by leukocytes. I intentionally indicate here yung alpha na sign, and it is used to spell the word leukocytes. That is to make you somehow understand that the leukocytes are the primary producers of interferon alpha. Interferon beta, on the other hand, is produced by fibroblasts, and intentionally highlighted the letter B's on the fibroblasts. That is to give you somehow an idea, or it will make it easy for you to memorize, that interferon beta is produced by fibroblasts. Both interferon alpha and interferon beta have antiviral activities. Therefore, they are classified as type 1 interferon. So is there such thing as type 2 interferon? Yes, there is. And it is interferon gamma. I know this one rings a bell because you have already heard it in our previous discussion. Now, I want you to take a look at the gamma sign in there. Okay, I just want you to imagine that both of his arms will be straightened to make it appear like letter T. Why? Because interferon gamma is produced by the T-delayed hypersensitivity. I hope you can remember that the T-delayed hypersensitivity functions to recruit more monocytes or macrophages and T cells into the site of infection. And that doesn't have to do with fighting of viral infection. That's why even if this chemical is called interferon, it doesn't have the same activity as interferon alpha and beta. So that's why interferon gamma is classified under type 2 interferon. Take note, interferon gamma is produced by T-delayed hypersensitivity, and T cells are classified under specific immunity. So of the three interferons, one is classified under specific immunity, while two are classified under innate immunity. And the interferon that falls under specific immunity because it is produced by a T cell is the interferon gamma. So this is just to help you recall that the T-delayed hypersensitivity, the blue-colored cell in the picture, will release the green-colored interferon gamma into the blood. And what will happen later on? The interferon gamma will recruit more macrophages and more T cells into the site of infection. The third line of defense involves now the actions or functions of your T cells and B cells. And we will have a separate discussion on this one on our next lecture. So let's have a review. You have three lines of defenses, two of which fall under non-specific immunity, and these are the first line and second lines of defenses. And then you have here the examples or immune responses classified under them. And the only line of defense that falls under the specific or adaptive immunity is the third line of defense. And listed under it are the immune responses mediated by T cells and B cells. The next question now is, if the immune responses are sequenced like that of how we play the chess game, how can the first line of defense will lead to the third line of defense? So I want you to imagine, I'll just make a short story. The skin is supposed to not allow the entry of microorganism, and that's first line. Should there be a break in the skin, what will happen is there will be inflammation. And inflammation will try to localize the infection. and there would be production of low levels of chemotactic stimuli, which will now attract white blood cells from the blood vessel, particularly our neutrophils, right? The neutrophils will undergo the process of chemotaxis, and once they are already out of the blood vessel, they will start engulfing the invading microorganism. And you know very well that neutrophils will only last for 1 to 2 days and they will die in the process called apoptosis. And they will be replaced by monocytes. And monocytes are not as good in killing as neutrophils. That's why monocytes will act as antigen-presenting cells. So at this point, we have already discussed first line of the skin, first line of defense of the skin, and the second line of defense involving the inflammation, chemotaxis, and phagocytosis. I want you to imagine that once that macrophage or antigen-presenting cell will present the antigen to T helper, you are now bridging second line going to third line of defense, because once the T helper will be activated, it will activate the B cell, which is also part of the adaptive immunity, and the B cell now will become plasma cells, and plasma cells will produce antibodies. Now you know how to link first line of defense to second line of defense, then into the third line of defense. And I hope that somehow my lecture made you create a summary or a connection between the different immune responses in our body. And that ends our lecture on Introduction to Immunology Part 4. Thank you very much.